The overall goal of this procedure is to target microinjections to the developing Xenopus kidney.
This method can help address key questions in the field of developmental biology, such as how the kidney forms and what genes are important for this process.
The main advantage of this technique is that it maximizes the delivery of microinjected reagents to a targeted tissue.
Visualization of this method is important because identification of the correct blastomere for targeted microinjection is critical.
To begin this procedure, both testes were isolated from a single male frog and placed in a 60mm Petri dish filled with 10 milliliters of testes storage solution. Then, store the testes at 4 degrees Celsius. Next, eggs were collected from a female frog in a 100mm Petri dish. Pour off excess water from the Petri dish. After that, cut off 1/4 of a testis in testes storage solution. Transfer the piece of testis to the Petri dish containing the eggs. Subsequently, cut the testis portion into small pieces. Add MMR plus gentamicin to the Petri dish to cover the eggs. Mix them by swirling the dish, and then wait approximately 30 minutes for fertilization to take place at room temperature. After fertilization, the pigmented side of the embryos